record this. Again, in college, most professors are not going to record their lessons. They're just not. If you miss a class, you missed it, which is freaky, right? Because you're paying $53,000 a year. You got a test next week. You don't have time to miss a class, but you're sick, or you got your mom sick, or you missed it because you slept in, or whatever. Now what? You got to find a way to get those notes from somebody. Again, back to your friends, back to your study group. Study groups have been shown, especially in professional school, study groups have been shown to be strongly correlated with success. All right? Study groups, not friend groups, right? Not, of course you can fool around and talk. I mean, that's part of being human. But it's about focusing on getting the tasks done that you're asked to do in class. So if you start to form study groups now, you'll see some of the more successful 10th graders, you'll see them in small study groups. They're studying after school. I've seen them. They're going over and reviewing material. Why? Because they want the Josie Edwards Scholarship. They want $500,000. They want to go to medical school, and they know it, right? So you have to find your colleagues that are of the same mindset, of the same kind of focus that you have, right? And what I found in my study groups in college and medical school is that when I'm teaching someone, when I'm explaining something to someone, and I successfully explain it to them, I understand it better. So it's not a bad thing that someone in your group doesn't understand. It is a bad thing if somebody in your group expects you to do all the work. And then that's no good. All right? You can't spend all your energy on other people. You have to spend a lot of it on yourself. So, but helping someone else can actually end up helping you. So consider that as you're in a study group, just because someone's not helping you helping someone else is helping you.
Pardon this interruption. We have contractors here working on the fire alarm system again today. So if you should hear the fire alarm, please disregard it. It is not an actual fire drill. Thank you.
So DNA, deoxyribose nucleic acid. That's what he talked about yesterday. And let's walk through some of the stories he said and kind of make the connections of what he was trying to make with you. There was a time when, well, let me start with what we know, because they knew something at the time. You know it as well. If you've looked at your handouts, you see that when in your biomolecule handout, you see that you have lipids, right? And the one lipid that we looked at was the triglyceride. I'm just sketching it out. I'm not going to worry about all the hydrogens and oxygens. I just sketching it out here. The triglycerides, some saturated, some unsaturated. Right. Remember, if it's a double bond, that will be on the test. So remember, if there's a double bond, it's unsaturated, right? We covered that, yes? We feel comfortable that you'll be able to tell the difference between saturated and unsaturated fatty acids. This one's unsaturated. This one is saturated. And the word for this is triglyceride. So thank you. Tri means three glyceride bonds, so 3 fatty acids.
So if you see like an equal sign, it's unsaturated?
The equal sign is a double bond, right? So it's sharing 2 pairs of electrons. So it's unsaturated. So if you see a double bond, yes, it's unsaturated. But let's not call it an equal sign because it's two. That's all right. You're right. It looks like an equal sign. But it just means that you're sharing two pairs of electrons.
Thank you.
Yes, bless you. So this was the example of lipids, right? And there's some others there. I think if you look, we know that they don't have a monomer. They don't have there's no single unit that you can repeat over and over again to make a larger molecule. We don't, fatty acids in a sense can be put together to make something bigger, but they're not really, there's no real polymer. There's really no polymer. So lipids don't have are not polymers. So the one biomolecule is not polymers. Then we looked at proteins. And proteins have two hydrogens, and they have a central carbon, and then they have this R group, and then they're connected to another nitrogen, right? Well, that's not true. They're connected to a carbon that then has a double bond, right? So when you have an amino acid, you have this going on. And when you connect it to the next amino acid, this is a monomer. This is a monomer, so this repeats. And this connects another amino acid. So these amino acids are going to become very important. And that's a carboxyl group at the end. So again, you have just to repeat what he had said earlier, that this is your acid part. It's a carboxyl acid. That's one of the functional groups you need to know. So these are both carboxyl groups. I hope you see that. And then you have an amine group here. So together they're an amino acid. Together they're an amino acid. Oops. So you have two amino acids when you're putting them together. Let me ask you a question. I think it was in that video that I had you watch for homework and extra credit, right? To watch the video and take notes on it. One of the things that you had to do is see, how do you put these together? The way you put them together is you have to, in order to bond these, to make a bond, carbon can only make 4 bonds. As 1, 2, 3, 4. So carbon's already is bonded to everything it can. Nitrogen can make 3 bonds, 1, 2, 3. So nitrogen's bonded to everything it can. How do we bond these two together? You have to get rid of something. That's right. I heard some people say it. You can only create a double bond or triple bond if there's room. There's no room here because, as I said, carbon can only make 4 bonds. How many bonds does carbon have? 1, 2, 3, 4. So that's full. Nitrogen can only make 3. 1, 2, 3. So it's full. So how do we bond these together? We got to get rid of something. So what happens is that in almost every polymer in your body, this leaves in the form of water. And when that leaves, if you watch the notes, you'll see that this is the case. When that leaves, what you're left with is, I'm just going to go ahead and redraw it here, a nitrogen, right? You have a nitrogen, you have 2 hydrogens connected to carbon, connected to hydrogen, connected to, again, that R group, connected to carbon, double bonded oxygen still there, so that's 3. The fourth bond, now the carbon is free to do what? To bond to what? To the other amino acid, to the nitrogen, right? And the nitrogen can bond because it got rid of the hydrogen. This carbon can bond because it got rid of the hydroxide. So now you can form, you still have a hydrogen there. That's a carbon. There's your hydrogen. There's your R group. There's a double bond O and the OH. So now you have just 2 amino acids. But guys, you can keep doing this for very, very hundreds of amino acids long. You've been doing this for a long time. You can have, I'm gonna go ahead and draw in just circles. I'm gonna draw just 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is just 10 long, but understand that these 10 can, it can be hundreds, okay? In fact, why don't I just make the point by actually just cut hundreds long. It doesn't have to be 10 or 2 or 3. It can be many, many long. And you take, all you got to do is remember that within each of these is a nitrogen. Oops. Within each of these is a nitrogen, hydrogen, carbon, hydrogen. There's an R group. There's a carbon, double bond, oxygen. But then there's that bond, because you got rid of water, you have that bond between the carbon and the next nitrogen. So when these are all bonded together, oops. These are all bonded together with those electrons, those 2 electrons being shared between carbon and nitrogen all along the protein. So they're linked in a chain. Do you see the chain? Like putting beads together. But the beads are actually amino acids. Now the interesting thing is that these proteins, these R groups, they can equal. This is an equal sign, but instead of using equal, because I just said we don't want to confuse it with a double bond, so let me go ahead and just put an R group can be 20 different groups. There's 20 different ones. There's big ones. There's small ones. There's hydrophobic and hydrophilic and acidic and basic. There's all kinds of different interesting chemistry. This makes them very interesting. They can do a lot. And the key word is do a lot. Very, 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 very functional. In fact, most of what you consider your body, your hair, your nails, your skin, right? The stuff that you see, most of it is proteins. If it's doing something, it's probably a protein because they're so functional, because they have so many different side groups. Like, you ever see a Swiss Army knife? The proteins are like Swiss Army knives. You just pull open a can opener. You can pull up a screwdriver, get all kinds of things. Even some have little hammers. So you can have all kinds of different things that proteins can do. Why? Because of these 20 different groups. Then we talked about carbohydrates, didn't we? We talked about carbohydrates. And carbohydrates, remember, are basically just C, H, and O in a 1 to 2 to 1 ratio. 1 to 2 to 1 ratio. Every 1 carbon, you get 2 hydrogens, you get 1 oxygen. We talked about that. That's an interesting property of carbohydrate. Mostly carbon and hydrogen and oxygen, that's a carbohydrate. And we drew a bunch of them. We talked about lactose. We talked about disaccharides. We called them saccharides. We talked about lactose. We did a whole little stories and examples of people that are lactose intolerant, correct? We talked about galactose. It's another one of those monomers. The monomers are monosaccharides, right, with carbohydrates. They're monosaccharides. And the monomers for proteins are amino acids, right? These are the monomers, the building blocks. You put them together and you get a protein. You put together a monosaccharide and you get polysaccharides. So what are the monosaccharides? The monosaccharides are this hexose thing. There's 6. Let's go ahead and, if you drew it, it would look something like you have an oxygen, you have a carbon, you have another carbon, you have 6 carbons. Oops. It's horribly drawn. Forgive me. I'm going to have to actually erase it. I'm sorry. I hate wasting time, but I just can't. So then each of those has hydroxides, but that would be a glucose molecule. A fructose has the same number of carbons, but it's bonded to the second carbon, so you have this carbon that's sticking out there, and what you end up with is a pentagon, even though it's still 6 carbons. They're both hexoses if you count them. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. They're both carbon, but this is known as fructose because it forms a 5-ring structure. It's going to act differently, right? Things that are round act differently from things that are square, right? You can't really use a square ring. You pretty much need a round ring. Fructose. I'm just going to have to use it like so. So that's one. Now there's another monosaccharide becomes very important. Is a 5, these are hexoses. I'm going to put the word hexose. Remember, ose means sugar. I'm going to draw now a pentose. What do you think pentose means? This has 6 carbons. What do you think pentoses have? That's right, 5 carbons. There's many different pentoses, but the ones we're interested in look like this. There's an oxygen, there's a carbon, there's a carbon, there's a carbon, there's a carbon, then there's a carbon. And now count them. There's 1, 2, 3, 4, 5. There's obviously an oxygen off of each of these, OH group. Remembering that each carbon needs a hydrogen, needs 4 bonds. Then we know that this has to be happening. So you know that each carbon has to have 4 bonds, so it's mostly carbon and hydrogen. If you count them, how many carbons, if you have 5 carbons, how many oxygens would you expect? 5, because it's 1 to 2 to 1. And how many hydrogens? 10. So let's count. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So 5 oxygens, 5 carbons. That's great. 1, 2, 3. Oh, I missed one. Do you see how I missed one here? Every carbon needs 4 bonds. So 1, 2, 3, 4, 5, 6, 7, 8, 9. I'm missing one. Where is it? This carbon only has 3 bonds. You notice how I caught my mistake there? If every carbon has 4 bonds, then I have to have 10 hydrogens. I have to have 5 oxygens. That's it. So this particular molecule is known as ribose. It's a sugar, so it ends with OSE, ribose. Now, interestingly, if we have the same exact sugar, notice I just copied it. Copied and pasted, that's all I did. Same exact sugar, and I got rid of this oxygen, so that all there is 2 hydrogens. Then it's not ribose. What is it?
Is it fructose?
See, I was wondering if somebody caught it yesterday because he talked about it yesterday. Without an oxygen, it's called D.
Is it
Say what? Say it.
deox?
Deoxyribose. So there's ribose, and then there's deoxyribose. The only difference, if we count them, remember how we count these structures? We count them 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. That second carbon, whether there's an oxygen or not, that tells you if it's deoxy or if it's ribose, or is it deoxyribose. It's just that second carbon, whether there's an oxygen or not. That changes the sugar. That makes a big difference, and we'll see in a minute. But those are monosaccharides. So there's deoxyribose. Here's ribose. Oh, I missed this guy. What? What's happening? No, I don't want that. Why did it do that? Anyways. There's ribose, there's deoxyribose, okay? So what's the other group that's left that we have to talk about? We got lipids, protein, nucleic acids. That's right. Now remember, these are polymers. By the way, you can make a polymer of these as well. Take a bunch of these, put them in a long chain, and you got that stuff that you sit on, those wooden stools, that paper you write on. It's called cellulose. Cellulose is a polymer of carbohydrates. That's found in plants. It's a polymer of carbohydrates. I'm not going to go into the details, but look, guys, all you got to do is make a chain of these monomers, which you can change out the monomers and change the quality of the polymer. But so if you used all glucose and you link them up one way, you get this thing called cellulose. If you link them up another way, you get something called starch. You can eat starch. You cannot eat cellulose, right? Nobody's going to sit here and eat paper. I mean, you might have known someone in elementary school did that, but no, people don't eat paper. At least they won't digest it well, okay? You won't be able to break it down. But if you eat starch, you know starch as potatoes and yams. That you can eat.
Don't starch affect your iron?
Say what?
Don't starch affect your iron?
I'm not sure about that. But I'll get back to you on it. Let's discuss it after class. Let's look at nucleic acids. That's the last group. And nucleic acids, what they are, before we get into what they are in the drawing of it, I'm going to start like he started and tell you what people used to think they are. When people started thinking about heredity, the word is heredity. The word inheritance comes from it. Or is related to it, I should say. Heredity. What do you think heredity means? You're right. In this case, it is the genes. But in general, it means something you get from the people that came before you. Your parents, your grandparents, your great-grandparents, what have you. So you're right. In this case, it would be genes. But back in the day, back in the 50s, they didn't know that. They're like, look, we know something gets passed. They called it the transforming. And you know what transforming is from the movie Transformers, right? Something that changes. The transforming principle. And they don't mean, and like you said in the video, they don't mean like the way we normally use principle. It's used in an antiquated way, in a way that we don't use it anymore. But they mean the stuff of inheritance. What is the thing that you inherit? Like, you know, you got your mom's eyes, your mom's nose, you got your dad's height, you have your dad's earlobes, you know, whatever. You know you got that. But what is it? How did he give you that? When you were in the womb, did your mom go in there with a little scalpel and a hammer and start pounding your nose in a certain shape? Right, right. We know that today because we taught it to you. You're right. Everything you're saying is correct. What I'm saying and what he was trying to say yesterday is back in the day, they didn't know. They were like, look, it has to be one of these 4 biomolecules. When we take a person, we grind them up in a blender, right? And we separate the pieces. We find four different kinds of molecules. We find carbohydrates, we find lipids, we find proteins, and we find nucleic acids. Which of these 4 is the stuff that gets passed on from parent to child? We didn't know at the time. They were thinking that most people thought DNA was too boring. It can't do a lot of stuff. It's not very useful. It's not like a protein. Protein can be an enzyme. It can be like your fingernails hard. It can be soft like your hair. It can be soft like your skin, or it can be hard like your fingernails, or a horn, right? Like a antler. Proteins can be all kinds of things because there's 20 different kinds that you can put together in all those different ways. DNA is boring. There's only 4 kinds. I'm about to draw it for you, but there's only 4 kinds, he says. 4 kinds of DNA. Yes. So why it doesn't make sense that DNA would be the thing you inherit from your parents. If you want to think about it this way, if you're going to write a book, someone volunteer their name for the sake of the video. Go ahead. So James. If we're going to make James, we're going to build James. We're going to start with a single cell. We need instructions, right? We need instructions to when we're building. Like, if you're going to build a house, don't you need instructions? Don't you need you need a blueprint. You need something to tell the plumber, look, put the toilet here, right? You don't want the toilet in the bedroom. That makes no sense. You need to have bedrooms here. I need you need a plan. Well, James had a plan. He got it from his parents. That plan was written. It's written in a language. It's a language. It's written, today we know it's written in DNA, correct? But back then, if you're going to think about writing a book as complicated as James, right? James is very complicated. He has 2 eyes. Each of those eyes are connected to a brain. They have all kinds of molecules that collect light. I mean, you created 2 eyes. They're so complicated. They can see light. And a brain that's so complicated, they can take that light and interpret it into a picture of what's going on. You have ears with all little tiny bones that pick up sound, and you translate that sound into something that makes sense. That's insane. That's insanely complicated. You take food like a steak or an apple or salad, and you break it down to the most smallest possible molecules. You absorb it into your blood, and then you make more of yourself. That's complicated. It's incredible what you're able to do. You have all kinds of organs. You can breathe air. You can run. You can swim. You can think. You can make tools. How are we going to make James? You need a complicated book. Don't you think the book is going to be pretty big? Let me tell you something. It's billions of letters long. If this were James's book, it would, and we stacked them one on top of the other, it would reach the moon. That's how big the information is that makes James who he is. And that's true for all of you. Because you're so complicated. So we need to know, now knowing that, would you choose, if you're an intelligent human being back in the 40s, would you choose DNA only has 4 letters, or would you choose something like proteins that has 20 different letters? Proteins. Why? I mean, we know it's DNA today. You're correct. But we would have guessed, and every intelligent person guessed proteins because they're so complicated. But it turns out, if you think about it really deeply, you have to be reflective. It turns out it is DNA, and their answer is kind of simple. You want that book to stay stable. You don't want, when you're making the copies, because you went from one copy of a book that goes all the way to the moon, and then you created 1 trillion copies, because you have about 1 trillion cells in you. And you created 1 trillion copies of those. And you're constantly making cells, so you're constantly making copies. The one thing you don't want is there to be a mistake in this message, right? You don't want all of a sudden James starts growing another head off his shoulder, right? That'd be kind of creepy and weird and wouldn't function very well. You wouldn't want James's anus to be out of his throat, right? Or his nose on his butt. That would not make sense, correct? It wouldn't work. You want to make sure that the message stays stable. You don't want it reacting. You don't want it changing like crazy. If you made it out of a protein, it could do a lot of stuff, but you would then, you take risks that you're going to change change it too much, correct? Like when you make your rings, why do we use gold? Gold's pretty boring. Gold is incredibly boring. Why do we use it for jewelry?
Because it's not that easy to change or break.
That's right. It's not that easy to change. I can stick this hand. I stick it in bleach all the time because I'm constantly washing things. I don't have to worry about this changing. If I made this out of tin, nickel, copper, even silver, it would change, wouldn't it? Yeah. If it was copper, it would turn green. And it's interesting. Isn't that interesting? It's much more interesting. It does a lot more. Copper does a lot more than gold. Gold is boring. It's not going to change. But it's going to last longer. And that's why DNA is the code of life, because you want it to last long for your whole life. In fact, you want it not just to last your life, you want to pass it on. And you want it to stay the same from generation to generation. You don't want humans to have 2 arms and 2 legs in your generation, and then the next generation have 3 arms and 3 legs, and the next generation you got 6 arms and 6 legs. That wouldn't be any good, would it? Like, that doesn't make sense. So we need to have something that's stable, like gold. Nice, stable, secure, that lasts a long time. Yeah. That's a really good question. People that are born with multiple heads are what we call Siamese. Well, they're not called that, but people call them Siamese twins. They are people that were two people growing together in a womb. They got too close, and the cells started sharing space and sharing messages. They were messaging each other, and they became, in a sense, one. Connected, strongly physically connected. They're still separate human beings. Their brains are separate. They think differently. They feel differently. But they might share a liver. They might share a hip. The lucky ones don't share a heart and don't share a spine. Because if you share a heart or a spine, then you can't separate. But if you share just a kidney and a liver, then and you have enough bone, then you could even get rid of one person could lose even a piece of the hip. You can replace it with an artificial hip. But if you share an irreplaceable organ, they're together for the rest of their lives, which isn't a horrible thing. There have been people that have been congenital. We call them congenital twins that are born that live their whole lives together. But imagine though the struggles that they would have to have. Like, it's something I can't even think about doing. But yeah, that does happen. That's not genetic, right? That's not a gene thing. That's a growing up thing. That's an environmental impact.
Well, like, what happens, like, 'cause I've seen, like, how these two people, their faces, like, connected, but they have different bodies.
Yeah, when their face, it's the same thing. They were twins. They got too close in the womb, and they started growing into each other. So what we can do today with our technology today is separate them. And we can reconstruct the jaw. We can do facial reconstruction. We can take tissue from the leg, for instance, and move it up to the face. You can do stuff like that and rebuild the things that were taken away, right? In the 50s, 40s, and 30s, that was not possible. So if you were born connected to the face, you stayed connected the rest of your lives. In most ancient cultures, they would be called, they would be holy. In a lot of cultures, they'd be holy. Some, they would have just killed them at birth. The Spartans would have probably thrown them on rocks. But they might have been considered holy people and people connected to God or whatever, just as many schizophrenics were considered holy as connected to God in some way. Yeah.
If that's what happens, like when you're born, do disabilities have something to do with genes? Like
they can. That is a really good question. It's something we need to think about. It's something kind of a contextual thing, because a lot of people have this misconception that every disease that babies have is something they inherited. That's not true. There's different reasons people have sicknesses. For instance, you could be born with something called cerebral palsy. It can impact the way you think. It can impact your body. Some people with cerebral palsy are impacted physically. Some are impacted mentally. My aunt was born with cerebral palsy. She was a twin, and she was born with cerebral palsy. She had problems with her thinking. She was physically fine, but her thinking was off. All right? That is not genetic. That is lack of oxygen in the womb. So if something happens and the umbilical cord gets wrapped around the neck, or oxygen somehow is during birth is the oxygen to the brain is impacted, then that person can be born with damage to the tissue. That is not genetic, right? That's another one of those environmental situations, just like the twinning that we talked about a minute ago. Vertigo, I'm not sure. I'm gonna guess. It's just a complete guess. Is genetic in some way. Now, is there environmental? A lot of diseases are both genetic and environmental. For instance, high blood pressure, heart disease. Heart disease, my father had heart disease, but he smoked, right? And smoking, we know, causes heart disease. But then but there's people that smoke their whole lives and don't get heart disease. Well, what's the difference? Well, their genes, for some group, makes them more susceptible than others. So it's complicated, because it's not all one-to-one. Sometimes one thing changes and it makes you more, what's an example? Okay, so skin cancer is caused by the sun touching your face too much, the UV radiation penetrating your DNA and changing it. However, people that are born with darker skin have protection against that, natural protection. People that don't have that darker skin don't have that protection. But you both can get skin cancer. Does that make sense? So one is more susceptible than another because of your genes. And that's true of a lot of diseases. You don't necessarily get the disease from your genes, but your genes can make you vulnerable to those diseases.
Wait, is diabetes genetic?
Type 2, we know type 1 is. Type 2 it runs in the family as well. There's a genetic component, but there's an environmental component as well. Eating too much sugar repeatedly, being overweight, causing your body to be inflamed, having inflammation, not getting exercise, not taking care of you for a long time can cause your genes, right? Because of your genes, cause you to be diabetic. Other people do exactly the same behavior and they never get diabetes. or at least they get it later in life. All right. So that is something that you need to consider, okay? But the transforming principle they thought was proteins, but it turns out it's DNA. Let's take a look at what it is. Do we have time? Well, I can draw this, the thing he drew, and then we're done. And by the way, great job today. Great job today. So oxygen is connected to carbon. We're going to draw a deoxyribose because we're going to draw DNA. So now when this is a deoxyribose because it has hydrogens here. I'm not going to draw all the hydrogens, okay? I'm just going to draw this because we're running out of time. Up here, of course, you have an OH group and an H, two H's sticking out here. Down here, you have also an OH and an H. Now, if we don't, DNA's monomer is not ribose, but it's something called a nucleotide. Now, when we take, and again, you have this drawing in your packet. If we take this carbon and we connect it to a phosphate, that's a phosphorus, connected to an oxygen, connected to 4 oxygens. That's a phosphate. To make it simple, this PO4, I'm going to draw it from now on. You're going to see me draw it like this. Is that okay with everybody? Because it's too many. You saw him draw ATP with all those phosphates. You all freaked out. You're like, what is that? How did he draw it so fast? What is that? It's ATP is what it was. So I guess we'll finish this tomorrow, but just know that right off here is the nitrogenous base. And that's called a nucleotide. Okay, so this is called the nucleotide. Let me give you a better drawing of it. Nucleotide. Nucleotide. And I'm going to write it in a minute. Let me just get a... There it is. So there it is. It's called the nucleotide. Let's take a look. I think it's better if it's drawn professionally. So it looks kind of crazy, doesn't it? Doesn't this look crazy to you? This is me lost in Parma, right? Because all the houses look alike. And if you're not paying attention, you're confused right now because you're like, what is going on? I don't know what this is. That's okay.
Oh, you can see the difference if you really look.
Yeah, it is. It is tough to look. So now let's take a look at this. Let's start with the nucleotide. This is a let me get the better pencil out. Nucleotide. What is a nucleotide? What are the 3 parts to a nucleotide? There's phosphate. There's a deoxyribose. There's a deoxyribose sitting here. Notice it's 5 carbons. It's exactly what we've been drawing now for 3 days. There's the oxygen, carbon 1. So this would be 1 prime, 2 prime. Let me do it a different color. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, 5 carbons. So it's a pentose, right? There's no, notice they didn't put the hydrogens, but they did put the oxygens. All the oxygens are there. Instead of having an oxygen here, you have this thing called the nitrogenous base on it. Okay? There's no oxygen on the 2nd prime. Why? Because this is deoxyribose, right? So it's deoxyribose, so there's no oxygen on the 2nd carbon. There's an oxygen on the 3rd one. There it is. The fourth one doesn't have an oxygen either. Well, the fourth one does have an oxygen. There it is. Okay. The fifth one doesn't have an oxygen because what's attached to it? Phosphate. That's the phosphate. So I'm going to go ahead and explain to you why there's no oxygen on them. That makes sense that there's no oxygen on them. So PO4 looks like this. It's a single bond to each one. Each of these normally have an OH group on it because so each of these is going to have an OH group on it. Now you got this is going to be bonded with the carbon on deoxyribose, right? So let's go ahead and draw deoxyribose. Let me fix my pencil. There's an oxygen that's connected to a carbon. I'm going to put the C, even though we don't put the C always, right? I hope you noticed whenever two lines come together in these drawings like you see here, what do we assume is there? A carbon. There's always a carbon at those connections of those lines. When you see these drawings, you have to know that there's a carbon unless they put another letter there. That's a carbon. Is that okay with everybody?
Yes.
All right, so I'm going to draw the carbon though. So there's a carbon. There's a carbon. There's a carbon. There's a carbon. And that carbon is connected to this. And then there's another carbon up here. All right, I'm not going to draw the hydrogens if you don't mind. Now, except for this last carbon, I'm going to go ahead and put everything that's there. So over here you have a hydrogen, you have another hydrogen, and you have an OH group. Now I got to bond this phosphate to this carbon. How do I do that? Carbon already has 4 bonds. What has to happen?
You get rid of something?
Have to get rid of something. I like that. So what's going to happen is this H here and this OH are going to leave. And what is it going to make? That's right. It's going to make water. Dehydration synthesis. These two are going to leave. Do you think you're going to see this on the test? Yes, you are. So these two are going to leave and make water. That's how we put things together in biochemistry, in life, in the chemistry of life. So we take a look at this awful drawing of deoxyribose. Again, I apologize. And now on this carbon that's sticking up here, you still got your 2 H's, but now you have also to this oxygen of the phosphate, you have, and usually these are negative under the conditions of so what you have is you have this connection that's made by, as you can see, by dehydration synthesis. We call this dehydration. This is review again. Hydration, meaning water leaving. Synthesis, meaning making something. You're making a bond by removing water. So now we added a phosphate to it, but we have to add the third part to it, which is the nitrogenous base. And I'm just going to put it NB here, but let's take a look at what it is. It's a nitrogen. You're going to connect to a nitrogen, this carbon to a nitrogen. But when you look at the drawing, remembering that, let me go down here. That's why I drew it up. Here you have 4 bonds, 1 H, 1 OH, and then here you have this thing called nitrogenous base. I'm just going to put an N here. That nitrogen has an H, and it's 3 bonds, right? Only makes 3 bonds. Nitrogen makes 3 bonds. I can't zoom in any further. What? Oh, zoom out like this? So now you're going to add the nitrogenous base to this. How do you make the how do you add it? If this has 4 bonds already, this has 1, 2, 3 bonds already. How are you going to connect these two? Get rid of something. What are you going to get rid of? You can get rid of the OH and the H, so water's going to leave. It's dehydration synthesis. So out comes OH and H. Out comes H2O, water, dehydration synthesis. And now you've made your nucleotide. You've made a nucleotide. That's how you make it in many, many steps. And this is actually the easy version. There's a lot of, when you get in organic chemistry and biochemistry, you're actually going to follow the electrons around. Yeah. Yes, you may. Dehydration synthesis means you're taking OH and H out, making water just like you did in class. You made water in class, correct? You're releasing energy when you make it. We know that. Yes.
This is the nucleotide.
Wait, I have a question in the back. Yeah. Oh, you have the same question. It's called nucleotide. I'm going to show you in a minute. I'm going to zoom in. I'm going to zoom back to the professionally drawn diagram that's in color in a minute. So I'm just showing you the connections and how they're made. Yeah. In a second. Just draw this. Do you have this? Everybody have this? Yeah. Yeah, this box, I'm going to show you what it looks like in a minute. But the nitrogenous base this is a bond to nitrogen, this is a bond to nitrogen, and then this nitrogen also has a bond to hydrogen. So this is just part of this molecule. I just drew as a box because I don't want to draw all those carbons. I'll show you in a colored picture in just a minute what it looks like. Nucleotides have nitrogenous bases in them. That is correct. That is correct. Just let me know when I can go, when I can move on. I heard I can go. Can I go? Trying to get this picture in here as well. Okay, so when we're talking about nitrogenous bases, let's zoom in. Let's go back. And this is a picture that kind of summarizes it all. And you guys can look it up anytime you like. But we'll look at what we're really going to focus in on is this part down here, this nitrogenous base. Here, that's connected to the 1'. So to the 1', you have a nitrogenous base connected to it. The 2' is missing an oxygen. The 5' has a phosphate. Okay. Now, when I drew a little box and I put NB, nitrogenous base, in the previous picture, there's the nitrogen. It used to have a hydrogen sticking off it. It also is bonded to a carbon. Remember, each of these is a carbon. Remember that the connection between each dot, each line rather, is a carbon, unless there's another letter there. So each of these is a carbon. Do you see that? And this carbon has how many bonds? 1. How many bonds does this carbon have? 1, 2, 3, 4. This carbon has how many bonds? 1, 2, 3, 4. This nitrogen has how many bonds? 1, 2, 3. This nitrogen had 1, 2, had 1 to hydrogen, got rid of the hydrogen, right? And now it's bonded to this carbon. This carbon has how many bonds? 1, 2, 3, 4. So it's the same rule over and over again. Every carbon has to have 4 bonds. Nitrogen can have 3, hydrogen 1. That's it. This carbon has 4 bonds. 1, 2, 3, 4. This carbon 4 bonds. 1, 2, 3, 4. This carbon has 4 bonds. 1, 2, 3, 4. This nitrogen has 3 bonds. 1, 2. Where's the third one? To hydrogen. They're just not. They tend not to draw the hydrogens. This nitrogen has 3 bonds. 2 to hydrogen. Correct. 1 to this carbon. This carbon has 4 bonds. 1, 2, 3, 4. This nitrogen has 3 bonds, 1, 2, 3. Do you see it? So you can apply that rule all over the place. It's there. Yeah? Oxygen can make 2 bonds. Look, every oxygen has 2 bonds. Look at them. Look everywhere you look. This one has 1 bond to this carbon. Where's the other bond? To the hydrogen. 2 bonds for hydrogen, for oxygen. 1 bond for hydrogen. Okay. So we attach the nitrogenous base here. This is a nucleotide in general, but there's only 4 nucleotides. What are they? Well, let me bring in a picture I just downloaded. These are the 4 possible nitrogenous bases. I told you proteins have 20 different R groups, 20 different variables, correct? This nitrogenous base can be 1 of 4. In DNA, it's only 1 of 4. There's a 5th one. This is found in RNA. All right. You have purines that have 2 circles, 2 rings. Are you clear on that? They're not really circles, they're rings. Rings meaning they go all the way around. They're not circles because they're hexagons or pentagons, right? But in any case, they're rings. They have nitrogen in them, correct? They're called nitrogenous bases. They are base. They act as a base, not an acid. Nitrogenous base.
You said there are 4 possible?
4 possible ones. Are you ready? I'm about to highlight them. There's an A, which is known as adenine. Or there's a G, which is guanine. Adenosine, adenine, or guanine. So of those, in that place, on that 1 carbon, you're going to find one of these 4 in DNA. Either adenine or guanine. What are they? They're both 2 rings, so we call those purines. All right. And then in the pyrimidines, you either have cytosine or you have thymine in DNA. So this is DNA.
So they all could be something.
DNA can have adenine, it can have guanine, it can have cytosine, or it can have thymine. That's it. Those are the only 4. Now, RNA, you've heard of RNA? It's ribonucleic acid. So instead of deoxyribose, you have ribose. They could have adenine. RNA can have guanine. RNA can have cytosine. But RNA cannot have thymine. It could only have uracil. It can only have uracil. So uracil takes the place of thymine in RNA. And the reason it does that is because of that missing oxygen in DNA. And you know, it's the only difference. What's the only real difference here between this one and this one? Who can tell me a difference? Can you tell a difference between uracil and thymine? What is the difference? There's an extra carbon in thymine. There's a carbon missing. There's a carbon missing in uracil, isn't there? Thymine has this carbon sitting out here, right? Uracil doesn't have a carbon out here. There's a reason for that. It's the amount of space. Literally, it's geometry. But we're not going to get into it. It's a matter of fitting, like trying to fit Legos together. This is literally what's happening here. You cannot fit uracil in the DNA. You can only fit thymine in the DNA. Yes. So everybody got that? Do you have to draw them? No, you do not have to draw them. Do you have to know their shapes? No, you do not have to know their shapes. Do you have to know that purines are 2 rings and pyrimidines are 1 ring? No, you do not have to know that. Look at that. I made your life a lot easier. You drew it good. Drawing it is not bad. You didn't waste your time. It's helping you find your way when you're looking at these molecules. But what you do need to, here's what you need to know by Monday. You need to know that DNA has an A, that DNA has a G, that DNA has a C, and DNA has a T. You have to know that RNA has an A, a G, a C, and a U. They do not have a T. Notice the color coding. All right. So that you do need to know. Again, you can get it from the video later if you need to. So it turns out that DNA shape that you all know that's in your school logo, right? The double helix. Right here. It's on the school logo. You have a double helix. Did you not know that? Yeah. Look at every bulletin ever sent to you. Look at every letter you've ever gotten from CSSM, and you'll see the double helix. So the double helix is, it goes from 3'. Now you know where that 3' is to 5'. We'll take a look at that in a second. It's actually 2 molecules. DNA, what they call a single molecule of DNA is 2 polymers, 2 molecules associating with one another. So that's what we call 1 molecule of DNA. It's actually 2 molecules. And each strand, they call them strands, stick together like 2 strips of Velcro. Like 2 strips of Velcro sticking together, DNA fits like that. It fits, and because it fits like that, it makes that shape. We have some kits up there. You're going to build them. All right? You're going to build DNA molecules here in just a bit, probably next week. I could do it today, but then you would have less review. In fact, I'll let you make that choice. We could build DNA molecules. I would love to. I would love to let you go at it. Here's the problem. You have a test on Monday. So you all have to make a choice. Do I go over the answers, or do you want to do the answers to your practice problems over the weekend? And you can work on the DNA molecules now. I will post the answers tonight. It's up to you how you want to handle that. Say what? We'll vote as a class. But let's go on and let me finish this idea here. So what do they look like when you look at DNA? This is the deoxyribose, correct? I'm going to draw out the nucleotide. It's a nucleotide, by the way, is the monomer. So we make the polymer. The polymer is known as DNA. So what does this polymer look like? It's made of a bunch of monomers. Let's identify the parts of the monomer. I'm going to identify 2 monomers here. Here's one monomer is a phosphate. That's part 1 of a nucleotide. There's a deoxyribose. That's part 2 of the nucleotide. And this is the base, the nitrogenous base. That's part 3 of the nucleotide. Which base is this? Which base is this that I just highlighted? Thymine. Look at it. It's labeled. If you can't see, let me zoom in, but I'll ask you to move. So there's part 1. There's part 1, the phosphate. Part 2 is the deoxyribose. and part 3 is the nitrogenous base. That's your nucleotide. But what is, if it's a monomer, we have to connect it to another monomer, correct? So we go ahead and we look below it. We see it's connected to a phosphate. Oops, sorry about that. We see it's connected to a phosphate here. That's 1. 2, there's a deoxyribose. And 3 is a base. So again, what do we got? 3 parts to this letter. 3 parts to this. 1, the phosphate. 2, the sugar. 3, the base. What base is this? Guanine. It says it, G, guanine. So this is T, and this is G. And it keeps going. 1 billion nucleotides. That's what's in your DNA. 1 billion nucleotides. One connected to the next, the next, the next, the next, the next, in a very specific order. That's what you call a gene. When you say you inherited a gene, you're talking about a specific set of A to T to G to C's connected by phosphates. Now there's another molecule on the other side. These are hydrogen bonds. We'll talk about those next week after the test. These are hydrogen bonds connecting these 2 strands. Now think about Velcro. You've all worked with Velcro, is that correct? You've never worked with Velcro? Never seen it? Never touched it? You're lying. I don't believe you. You live in the United States of America. There's no way you've never worked with Velcro. I can't believe it. I just can't. Huh? You had to have seen it. Oh, so you did see it. So you lied. Okay. Okay, I'm just messing with you. I'm just messing with you. Come on. So I knew you've seen Velcro before. So you have Velcro. You have 2 strips of Velcro, right? Yeah. Is it easy to take a single strip and separate it and break it up? No, you have to use scissors to cut it, don't you? It's hard to cut a strip of Velcro. Oh, yeah. Like a single strip, right? But then you take those 2 strips and they stick together, right? It's easy to separate them. So there's a weaker connection between the 2 strips than there are between the covalent bonds of the strips themselves. Correct? Yeah. Well, that's the same with DNA. In DNA, you can separate the 2 strands easily because these are like the connection between the Velcro strips, where these are the Velcro strips themselves. So make that connection in your mind that we can separate the 2 strands easily. We cannot separate these parts easily. All right, so these 2 separate much more easily than each individual strand. So we have a nucleotide on the other side. Let's see if we've learned anything. Let's try to identify the 3 parts of the Velcro strip. So we have, let's look and find, this is upside down. Do you see how they're upside down from each other? This is in English, they call this foreshadowing. If you're paying attention, you're going to see this again. All these comments I'm making, you're going to see them again next week, especially when we build those molecules. These are upside down. We call this upside down nature anti-parallel. They're parallel, but they're upside down, so we call them anti-parallel. Okay? So here is part 1. Here's part 1. The phosphate, correct? Yes? No? Maybe? Okay. Here's part 2, the deoxyribose. Do you see that it's upside down? You see that here the oxygen is pointing up, here the oxygen is pointing down? Okay. And then the last part is the nitrogenous base, part 3. By the way, the reason it's upside down is the only way to make them fit. We got to fit them together. It's the only way they fit. When I say we, I mean nature. So here you have 1, the phosphate, 2, the deoxyribose sugar, 3, the nitrogenous base. Now we have another one and by the way, which base is it? Which of the four is it? A. See, it says A right there. A. Do you see that? Everybody see it? Somebody have a hard time seeing that this is an A? So A hydrogen bonds with what? A with T. Always A with T. Always A with T. In DNA. In DNA. In RNA, A binds with U. Hydrogen bonds with U. Hydrogen bonds. Now, we look at the next one, and we look at, this is connected. This is connected covalently again. There's a phosphate. There's a deoxyribose sugar. And here's a base. So again, to remind you, here's number one, the phosphate. Here's the sugar. And there's the base. 3 parts to this nucleotide. Which nucleotide is this? C. C is bonded to what? Hydrogen bonded to what? To the G on the opposite strand. So G always bonds to C. If you see a C on one, you're going to find a G on the other. They don't fit any other way. They just don't fit. So if you put a T over here, the G would have to change. I'm sorry, the C would have to change to an A, or you'd have to change the T because they just won't fit together. All right. So these are nucleotides. That's how they fit together. You'll be putting together yourself. Now, I'm going to go ahead and talk about, the last thing I want to talk about with DNA structure is this double helix. Do you know what a helix is? Spiral staircase. A spiral, is it? A helix is a spiral. It's a spiral. It goes around and around like one of those circular staircases that go up to the second floor, right? It's called a helix, a spiral. All right. So when you have one strand, a bunch of, you're going to connect, I'm going to put a P, okay? I'm just putting a P. I'm going to keep it simple. Too late, right? And it's connected to a sugar, the deoxyribose sugar. Remembering that each of these, and of course that's connected to nitrogenous base. I'm keeping it very simple. Do you notice how simple I'm keeping it? I'm not putting the O's, the H's. I'm not drawing out the base. I'm not even drawing out the phosphate. It's just a P. I'm not even putting all the C's. I'm just putting this one C. Here on the third carbon, this is 1 prime. This is 1 prime. This is 2 prime. There's no oxygen there, but I'm going to go ahead and let it go. Here's 3 prime. Here's 4 prime. And there's my 5 prime. So 5 sugars. On the 5 prime, I find a phosphate every time. Every nucleotide, I have a phosphate here. So if I have a second nucleotide, if I have a second nucleotide, I want to connect them. Now I connected them, we made a nucleotide, but now we got to make a connection between this one and that one, don't we? Because these are all connected in a chain. We know that this OH, there's an OH group sitting off here. We know that there's an OH group sitting off here. I'm not drawing all the rest of it. What can happen? What has to happen if I'm going to connect them? That's right. You got to take these out, and you get H2O. That's how you're going to connect them. Dehydration synthesis. It's always OH and H. Just let's think of it that way. To make water. That's what we do. That's what's done. In all life on planet Earth, plants, bugs, insects, bacteria, humans, all build it the same way. So dehydration synthesis. So that means that OH and H go away. So I'm going to go ahead and get rid of it. There they go. Bye-bye. Out comes water. And now now what is it? Now you got this one. It twists a little bit. And you make a connection between those two. And you keep doing that. Billions of nucleotides long. So I'm gonna do something real simple. Are you ready? Yes? Yeah, I'm even gonna make it even simpler. I'm gonna simplify even more. I'm just gonna do this for the base. I'm gonna stick up a phosphate. I'm gonna stick up here nitrogenous base. It's just simple geometric shapes, if that's okay. Is everybody okay with that? Do you know what I'm talking about, though? If I draw this, you're not thinking, wow, why is it so simple, right? I'm drawing it simply so that you get the idea that what we can do is we can put them all together in a very, very long chain. Very, very long chain. All right? That would be DNA. 1/2 of the DNA. Yeah, you weren't looking at the screen and then you looked up and then you see that and it shocks you. So we can make a covalent bond between each of these, you see it? Between the 3' of one carbon of one chain and the 5' of the other, you can make these covalent bonds. These are covalent bonds, which means what? Are they weak or strong? Strong. These are strong bonds, just like the Velcro strip that's made very strong. Yep, Velcro has to be strong, or else it just does no good if it falls apart. We can connect them all. A DNA molecule is connected by a sugar-phosphate bond. Do you see this? Sugar and phosphate are bonded together. Y'all, you seeing this or not?
Yes.
So you see that they're all connected with a sugar-phosphate bond. A sugar-phosphate bond is connecting each individual strand. So each strand of DNA is connected to itself by sugar-phosphate bonds, put together by dehydration synthesis. There's 2 strands. This side here, please pay attention to this because this is going to come in handy later. This side here is the 5' end, correct? I can number them again for you. Oops. So here the sugar, that's the 5' end. What's on the other end of this molecule? That's right, 3'. Somebody's been paying attention. This end is the 3' end. So on one side is the 5' end, on the other is the 3' end. This side's upside down, so what is over here? 3' end, and down here is the 5' end. Because they're upside down. Now, in between these, you get the weak connection. These are 2 Velcro strips. When they connect, they're going to connect weakly. Yeah. Yes, they are upside down. They literally are upside down. This is the only way they fit. In between the nitrogenous bases, you get these weak little bonds. I'm calling them weak, but, you know, they're weak. Weak bonds, attraction, like magnets between these. Hydrogen, we call them hydrogen bonding. We'll talk about them next week. But they form. They start to pull each other together like Velcro pulls itself together, correct? I don't know if you've ever seen work with Velcro. I know some of you have not worked with Velcro. But when they get close to each other, they stick to each other. It's kind of annoying. And if you ever have a silk tie, gentlemen, if you ever have a silk tie, keep it away from Velcro. Or ladies, any silk. It just sticks like crazy, ruins your silk. I've ruined many a tie because of, as teachers, we're dealing with Velcro and we may be wearing ties. So they start to stick to one another. Does this look like a zipper to you? Yeah. Remember that, because to me it looks like a zipper as well. So when they come together, there's 3 dimensions. I'm drawing it in 2 dimensions in length and width, but there's the depth as well. And so when they start to come together, they start to form this. They start to do this. They twist and turn. Twist and turn. 2 strands twisting and turning together all the way down. And they twist and turn, and every time they twist and turn, they form this thing. You've seen this before. This is a double helix. 2 spirals. Double, that's on CSSM, that's right. It's a double helix. 2 spirals. Hydrogen bonded to each other. Hydrogen bonded to each other. That looks like a ladder, doesn't it? Like a spiral ladder. Again, she's just being obstinate. It does look like a ladder. So it's a double helix. This is, again, if this is 5', then you follow it along. And on this end, it'll be 3'. If this is 3', then it has to be 3'. You follow it along. This will be 5'. They're anti-parallel. That's the DNA shape of, by the way, this part here, these nitrogenous bases, they can be what? What can they be in DNA? What can they be in DNA? The nitrogenous bases can be what in DNA? What could they be? They are, well, hold on. Yeah, somebody just said it. That's right. A, G, C, or T. Right? Here, let's be clear about something. Now, somebody said something that kind of concerns me. This, I'm putting it in a dotted line. This is a nucleotide. This is a nucleotide. They are monomers. They're one. Put them together in a long chain, you have many. So we call it a polymer. So there's 2 polymers here. One strand is a polymer. The other strand is a polymer. They come together and they make a molecule of DNA, a double helix. Now, the nitrogenous bases change, don't they? A, T, G, or C, all the way down. Your series of letters is different from mine. That's why we're different. Your strand of letters are different from mine. Very, very small differences. Instead of a T, you have a G. Instead of a C, you have an A, whatever. Right? But those small differences add up and they make you a girl, and me a boy, make you your parents' child, make me my parents' child. Does that make sense? So that difference in the DNA. So this part here is different, but you see, this is all the same. Everything is built on this, on these 2, isn't it? Everything is built on the phosphate and the sugar. Do you agree? That's what's connecting the whole thing together. That's what's holding the nitrogenous bases there. That does not change. So because of those 2 aspects of it, we call this the sugar phosphate. Good. Yeah, their sugar phosphate bonds is correct. Backbone of DNA. It's not a bone. There's no back. But it does kind of look like a backbone, doesn't it? Have you ever seen the vertebra? Do you want me to show you what a backbone looks like?
Yes.
Yes? Hold on. Okay, so really quickly, take a moment. Take a moment and look at this. Shh. Look at this. Think of what one of these looks like, right? Look at it. Just take a moment and look at the screen. I'll wait for everybody to be looking at the screen. Look at the screen and take a look. Sugar and phosphate. Look at the sugar phosphate. The nitrogenous base has changed. Sugar phosphate doesn't. Look at the phosphate. Now look at a backbone.
It look the same.
They look very similar, don't they? That's what. Are they the same? No, they're not the same. There's a lot of differences. Yes, I get it, but they look similar. It's in your neck. So your vertebra has different. This is your neck called the cervical vertebra. This is your thoracic. So it's from the neck down to the abdomen. And then lower than that, we call it the lumbar region, lumbar vertebra. Yes. Really good question. Yes. Each strand has a sugar phosphate backbone. Do you see it? Sugar, phosphate, 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 sugar. You're looking at me like that because you're not looking at the screen. Sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way down. So we call it the sugar phosphate backbone. For each strand, look at the other one. Sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way down. They're the backbone of each individual strand. When they say a molecule of DNA, though, most of the time, most people think of a double helix or double strand. But to be absolutely on point, really, a molecule of DNA is one strand. Because a molecule is covalently bonded. These two are not covalently bonded to each other. If you want to get technical, each strand is its own molecule. By the way, those things you call chromosomes, you know those things you call chromosomes? There it is. This is a chromosome. There's some more parts to a chromosome, some extra parts to a chromosome we'll look at. But when you talk about chromosomes, you're talking about a chunk of DNA. So what happens if there's there are problems. We'll talk about that, but we're not ready for that yet. But yeah. Down syndrome is one of many options, one of many negative outcomes. Yeah.
How does RNA look different than DNA?
What a perfect question. Is it time to go?
No, it's at 10:07.
What do you mean by almost? For the record, as a student, so 12 minutes is almost to you. Okay, just trying to get a grasp of what you're saying. 12 minutes to me is a lifetime. So could we try to focus and let's answer some questions before we go? By the way, great session today. The rest of the answers to the home, I'm gonna have to post for homework the answers. You should be able to answer everything on your own, but I'll post the correct answers on YouTube tonight. I'm gonna do it during second and fourth, and I'll post them tonight. So you should be able to check your answers and make sure everything's right before the test. Okay? Now somebody asked a question. You know what? It's such a great question. I'm going to answer it before you go. I'm going to spend a couple minutes here answering it. What is the difference between RNA and DNA? And the difference between RNA and DNA turns out to be pretty straightforward. RNA has something called ribose. That means that sugar has that oxygen. DNA has deoxyribose. This will be on the test, yes. It's as easy, so you should be happy. It's as easy. RNA has A, U, G, or C. DNA has A, T, G, or C. Those are the 4 possible nitrogenous bases. RNA only has 1 strand. It's a single-stranded. DNA is double-stranded. DNA's only job is to keep the That's right. Keep the inheritance is correct, but I'm going to phrase it a little differently, but you're right. Inheritable message. That's it. It has a message. Information. If you want, I can put information. It keeps it safe. That's it. The stuff that you're going to give your children, the stuff you got from your parents, is all in DNA. That's the only thing DNA's job is to do. We don't know the difference yet, but it's also in eukaryotes. You don't know this word yet, but you'll learn it. Cells with a nucleus. Some of you may know it already from middle school. So DNA is only found in the nucleus. It's only found in the nucleus. Is that clear? RNA, on the other hand, can act as a messenger. Yeah. E-O-U-S. Nucleus.
No, I'm talking about for the word, the E word.
Oh, E-U-C-A-R-Y-O-T-E-S. You want to put a K? You spelled it with a K? There you go. Eukaryotes. I don't care, K or C. Some people spell it with a C, but that's okay. We're moving on. Yeah. Adenine, thymine, guanine, cytosine. The 4 nitrogenous bases. So RNA acts as a messenger. RNA can act as an enzyme. RNA can act as a trait. I don't want to say it, so I'm going to leave the third function off. It's going to confuse you if I say it. I'm already pushing it by using the word eukaryote. All right, so I don't like using words I haven't defined. You know what I'm saying? RNA has some other things going on. We'll talk, but it can be used as a messenger. It can be used as an enzyme. So RNA is very interesting, but DNA's whole job is to hold your message. All right, great job today. Yeah, we can use DNA. We can use RNA to do a lot, and we do. We use RNA. We use some of our vaccines. We've used the RNA to make them, so they're very interesting.